Hi, everyone. So when we left off on our chapter 2, part 1 video, I was telling you something about the different types of molecular bonds. And what I'd like to do now is focus in on the polar covalent bonds that we find in a water molecule. Now, just to kind of pick up where we left off, remember that oxygen kind of hogs, I guess is the best way of saying it. It hogs all the electrons. You can see here in this diagram that the concentration of electrons around oxygen in a water molecule is really situated around this oxygen nucleus, right? And the electrons are kind of pulled away from the hydrogens here. The effect of that is that all the negative charge tends to, well, not all of it, but most of the negative charge tends to reside around the oxygen, while the positive charge tends to be up here with the hydrogen because you have a proton kind of sitting off, not quite by itself, but kind of off by itself. And recall that what I was telling you was that what we say is that oxygen is an electronegative atom because it will sort of gobble up your electrons, right? Now, what is the upshot of this situation where they're sharing electrons, so it is a covalent bond, but they're sharing these electrons unevenly so that you have a negative and a positive end. Well, I was telling you last time that what the effect of that is that if you look at these individual water molecules, what you find is that they act like magnets with one another, whereby the negative oxygen tends to kind of be magnetically drawn to the positive hydrogen. And you see that here and here and here. And where we find these hydrogen-oxygen magnetic interactions, those are called hydrogen bonds, okay? Now, one of the focuses of this chapter is in talking about water as one of the chemical foundations of life. And it's these hydrogen bonds that really set up the conditions that make water essential for life on this planet. So let's take a look at how some of that might work. All right, so I had to do some re-recording. Let me get rid of the things that I'd already drawn on here. All right, so what are some of the effects of water's polar covalent bonds, or its positive/negative nature? Well, one of the first things that we find is that water acts as an excellent solvent for other polar molecules, okay? So let's take a look at what's going on down here. What we have is an image, these little frog head looking things, those are your water molecules, okay? So we've got a negative charge on the red oxygen atom and positive charges on the white hydrogen atoms. And then what you see here is this big green atom is supposed to be a chloride ion, and the positive is supposed to be a sodium ion. Let's go back and look at the sodium chloride one more time. I had shown you this in an earlier slide. So up here we have sodium chloride, which you probably know as table salt, right? And just as a quick refresher, this is the valence shell for sodium. Sodium happens to have a atomic number of 11. And so it's got one, on its outer shell, it's got one lonely electron. But remember that the outer shell wants to have 8 electrons, right? So it's only got one. This is pretty unstable situation. In contrast, chlorine, I should say, what is its atomic number? Its atomic number is, hold on, give me a second. Let's see, up here. 17, okay, great. So going back down here, if you were to work it out and do one of those diagrams that I was doing earlier, what you would find is that results in a valence shell that has 1, 2, 3, 4, 5, 6, 7 electrons, but then it's got an empty parking spot. This is a very electron-hungry atom. It is highly electronegative, just like the oxygen was, okay? So what it will often do if it's in association with sodium is it will snatch this rogue electron off entirely and keep it for its very self, that results in a sort of naked outer valence shell for sodium and a net positive charged sodium ion. And it results in an overall negative charge for this rogue chloride ion here, right? And remember, this is an ionic bond because they are not sharing, no share here. They've actually completely lost or gained an electron. Now let's go back to my water example. Let's see here. Down here. Okay, so you can see that because we have these partial negatives and partial positives on our water molecules, it sets up a perfect environment for these negative chloride ions to hang around, and they're just going to be surrounded by some of these positive hydrogens. And then you have a positive sodium ion, and it's going to be surrounded by some of these negative, positive sodium ions surrounded by the negative oxygens, okay? So you can see that it's going to work nicely for this to sit snugly in this water solution. So water is an excellent solvent, but it's only an excellent solvent for things that have positive and negative charges in it. And we call those polar type molecules, another word for them is hydrophilic. Hydro, of course, referring to water. And philic, you may know, is a word root for love, like you may remember from history that Philadelphia is the city of brotherly love. So hydrophilic compounds are water loving. Later on, we'll be talking about hydrophobic compounds or water hating compounds. Those are nonpolar and do not dissolve in water. But okay, that's one feature. So number one, water is an excellent solvent for polar compounds because of its polar covalent bonds. Number two, because water has this polarity, it really resists changes in temperature. So it's hard to change. And I'm going to use the symbol here. This is the Greek symbol delta, and it's used a lot in scientific notation to mean change. So hard to change temperature. Now that's not quite what I wrote up here, is it? What I said was it has a high specific heat or a high heat of vaporization. So let's look down at our notes right here. All right, what do those terms mean? Specific heat is the amount of energy required to raise 1 gram of a substance by 1 degree. Heat of vaporization is the energy required to change 1 gram of a liquid to a gas. So both of these refer to the ability to change the temperature of water. Now, what's happening at the molecular level when you change temperature? Well, temperature is really a measure of kinetic energy of a solution. What the heck does that mean, Dr. Jones? Kinetic, and this is my abbreviation for energy, G, okay? So it's the measure of kinetic energy. Kinetic energy is just the energy of motion. When you heat up molecules, they tend to like zip around, right? They move more. And when you take the temperature of something, what you're doing is you're measuring how quickly the molecules are moving, okay? Now, because water has these magnetic attractions, now magnets, of course, they're not permanently bound to one another. You can break magnetic bonds and reform them, but it does take some energy to do that. So because these things are magnetically bound to one another, it's quite difficult, or I should say it takes a lot of energy to heat this thing up, all right? So feature number 2 is that because of water's polar covalent bonds, because of its polarity, it's very difficult to change the temperature of water. The third thing that I want you to know about water is that because of its polar characteristics, it is less dense when frozen. Now let's think about that for a second. You guys know that when things are cold, they tend to contract, right? And again, remember that temperature is a measure of the kinetic energy of a solution or gas or whatever. So if we take the temperature of this and it's going down, going down, going down, that means that these molecules aren't moving very much. And what happens when you cool down gases or liquids or whatever is that the molecules tend to like condense and get closer to one another and kind of almost congeal until you have a solid, right? That's how that works. Well, that does happen in water. As you cool it down, the water molecules, they slow down, they start to congeal and kind of get closer to one another and closer and closer and closer. But then when they get to the freezing point, a very interesting thing happens. You guys have probably all had the experience of holding two magnets together, but holding like the positive end to the positive end. And what happens? Well, the positive, if you hold positive to positive on magnets, they repel one another. Similarly, if you hold negative to negative, they also repel one another. So what happens when you freeze water, see we have these negatives here, negative to negative is repelling one another. Negative to negative is repelling one another. So once you get to the frozen state, there is a lattice that is formed that makes ice porous, that's P-O-U-R-O-S, at the molecular level. And so what that means is that it's less dense when it's frozen, and therefore frozen or solid water actually floats on top of liquid water, okay? That's not true of most other substances. So what I want you guys to think about is how are these things important to life, okay? Why are these features important to life? And I'm gonna to life. And this is gonna be a topic for conversation on Friday. So how do these three features sort of, how did they like set up favorable conditions, I should say, for the evolution of life? Think about that. All right, again, I had to re-record. Let me get rid of some of this. Boop, 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 boop. Okay, so two other features that I want you to think about, and thinking about the interesting attributes of water that result from its polarity. So we talked about three things on the last slide. Let's talk about two more, adhesion and cohesion. What is adhesion? Well, adhesion is the ability of water molecules to sort of magnetically stick to other molecules. Okay? So let's look at this glass test tube here. And can you make out that there's got water in it and it's making a bit of a U shape? This U shape is actually called a meniscus. Incidentally, if you're ever measuring something in the lab, you want to measure from the bottom of the meniscus. So why does water form this U shape? Can you kind of see this going bloop like that? Why does it form this U shape? Well, let's take, and we've got this little blow up here, so let's have a look at it. Well, what's happening along the walls is adhesion. And you can see here that we have the hydrogens, which remember those are partially positively charged, and they are actually kind of magnetically attracted to the glass walls of this test tube. And that's because the glass is slightly negatively charged. So we have another magnetic attraction. And we have this uphill slope. Well, why do we have this uphill slope? Well, the uphill slope is the result of cohesion, which is the ability of water molecules to sort of magnetically stick to other water molecules. So if this is kind of sticking to the glass, then what it's doing is this water molecule is sticking to this water molecule and it's pulling it up. And then this water molecule is sticking to this water molecule and it's pulling it up. So why are these two features important? Well, it has to do with the way the plants work, okay? So let's think about a plant. I'm gonna draw a really beautiful plant for you. Beautiful artwork here. Okay, so these are supposed to be the roots underground. Okay, bloop, bloop, bloop. And then up here, we're gonna have a leaf. Beautiful. And then, oops, a leaf. Okay, gorgeous. Beautiful, beautiful plant artwork. Okay, now somehow we have to figure out how to get water from the ground down here up to the leaves up here. And that water is transported in vascular plants through a network of tubes called xylem. Think of these xylem tubes, kind of like the veins and arteries of your circulatory system. So we're going to use these xylem tubes to get water from down here all the way up here. But how are we going to get water to go up, to defy gravity? Well, in your body, the way we get blood to move around, water and plasma and blood move around, is we pump it using a heart. But obviously plants don't have a pump, right? So how do they do it? Well, if you've got some sunshine and some wind, so I'll draw this to be wind, what's going to happen is some of the water molecules in this leaf are going to evaporate out. They're actually going to escape and become gas, right? And because the water molecules stick to each other via cohesion, they kind of hold hands. So as one leaves, it pulls another one up, okay? Which pulls another one up, which you can propagate all the way, oops, another one up, another one up. And so you can kind of see how that would work. The other thing that helps this process along is that as this water is being pulled up these xylem tubes, the water molecules adhere to the side of the tubes via adhesion. So this is how plant roots and plants in general are able to defy gravity and transport water up through a plant without the benefit of any kind of pump. All right? Let's talk some more about the chemistry of water. So, all right. Let's talk a little bit about water as it's just hanging out. Let's talk about some pure water right now. And when you have a solution of pure water, what will happen to a very small number of the water molecules is they'll actually dissociate into two different ions, a hydroxide ion, or an OH negative, and a hydrogen ion, which is an H positive. And if you recall, I'm just going to go back for just a second. Hydrogen ion is really nothing more than a proton. I think I showed you guys that up. Where did I show it to you? Let's see. Oh, here it was. Okay, so you can see a hydrogen ion is really just a proton, right? It's lost an electron. Boom, there it is. Okay, so back to where we started. Okay, so there's going to be just a small number of these water molecules that are going to dissociate into these hydroxide or OH minus ions and the hydrogen or H plus ions, okay? And these two arrows mean that this reaction is at equilibrium. So some of them are going to dissociate, some of them are going to reform, and it's going to go back and forth until it reaches a steady state. If you happen to have a solution that has more of these than it does these, then we're going to say that that is an acidic solution. And conversely, if you have a solution that has more of these than it does these, we're going to say that that is a basic solution. So when you're talking about an acid, what you're really talking about in acids, the acid right here, what you're talking about is the number of hydrogen ions, and what you're talking about when you're talking about a base is how many hydroxide ions or how few hydrogen ions you have is another way of thinking of that, okay? If you have equal numbers of both, then that's considered to be a neutral solution, all right? And so you guys probably know that we measure acids and bases using a scale called the pH scale. So let's talk a little bit about that pH scale. First of all, let me, one more time. Y'all, I had a mistake and I had to start over, which is a pain in the butt. Okay, so pH scale. First of all, why would we care about knowing how acidic or how basic a solution is? Well, let's go back down to my notes for you down here. Those positive and negative charges that you find in hydroxide ions or in hydrogen ions can mess with cellular chemistry, right? So remember that the hydrogen ions are positive, the hydroxide ions are negative. That can really mess with the chemistry that's going on in your cells. It can mess with the rates of chemical reaction. It can change the shapes of molecules. It can alter the way molecules bind to one another, and it can change the ability of molecules to dissolve in water. So it's kind of important that cells maintain a steady pH and that they have an ability to regulate that. And this is why we're interested in measuring pH. Now, you may be wondering where this term pH comes from, and it's actually from the French for power of hydrogen. I think the French word, I don't speak French, but I'm going to look at it for you. Hold on, I've got some notes here. Come on. Okay, so pH stands for power of hydrogen, and in French, I think it's actually puissance de hydrogène. I won't make you remember that, but I just kind of wanted you to know where it comes from, because if you're like me, you maybe wondered why pH is always written with a lowercase p, but an uppercase H. The H is the chemical symbol for hydrogen, which is always capitalized, but the little p is just for the power part. So that might help you remember where that comes from. pH, what it's really measuring is the concentration of hydrogen ions in a solution, okay? So if a solution has a lot of hydrogen ions, we say that it's acidic, but the fewer hydrogen ions it has, the more it has like a negative OH component, and therefore we say that it's basic, okay? If we look at where this comes from, if you were to actually look, say, at a solution of water, just plain pure water, which is said to be neutral, if you could actually measure the concentration of hydrogen ions in a neutral water solution, what you would find is that it was equal to 0.0000001 molar. So molar, or molarity, molarity is just the number of moles per liter, and it's something that you would learn in chemistry. Don't worry about that too much if that throws you off. Just know that it's a measurement of concentration. So we've got 1, 2, 3, 4, 5, 6, 7. Neutral is 7. So how do we get that? All right, here's how we get it. This is a really tiny, tiny number, right? And another way we can write tiny, tiny numbers is using something called scientific notation. So you would write this as 1 times 10 to the negative 7th, right? This is 10 to the negative 7th. 1, 2, 3, 4, 5, 6, 7. So that's how tiny like the amount of hydrogen ions is in neutral water. But that's just a crazy thing to write. So to make this scale a little easier to interpret, we do something called the logarithm. We actually take the negative logarithm. So the negative log of that right there, or this right here, is 7. So that's how you get a pH of 7 for something that is neutral, okay? So, but what that means is that as you go down in pH, you actually have more hydrogen ions. Let's take a look at this one. This is 1 times 10 to the negative 1, right? Which is the same thing as saying 0.1. Can you see that this is actually a bigger number than this is? So something that has a pH of 1 has more hydrogen ions in it than something that has a pH of 7. Therefore, it's more acidic, okay? I want you guys to be aware of that, because sometimes that's counterintuitive. But the lower the number, the more acidic it is. The higher the number, the more basic it is. The other thing that I want you to recognize is that because the way the scale works, every step on here is actually a 10-fold change, okay? So an acid that is a pH of 1 has 10 times more hydrogen ions in it than an acid that's a pH of 2, which has 10 times more than an acid with a pH of 3, etc., okay? So as you move up and down this pH scale, if you're going from like a 7 to a 6, that's a lot more acidic, okay? So if you really understand this, then you would understand what would happen if I had some black coffee in the morning and I'm like, oh no, I have to have my coffee, but I don't want to drink it black. I'm going to add some milk to it. So what would happen to the pH of my coffee if I added milk to it? Would the pH go up or would the pH go down? Okay, let's talk about that on Friday. Answer that question for yourself. All right, now I mentioned that acids and bases, particularly acids, the number of hydrogen ions, can really mess with your cellular chemistry, okay? So cells really try to regulate the acidity of their environments, okay? And they do that using something called buffers. So what is a buffer? It's a compound that minimizes the effect of adding acids or bases to solution. I like to think of it as a sponge, okay? It is something that can absorb hydrogen ions and keep the acidity of the solution fairly constant. And similarly, it can absorb hydroxide, negative ions, and keep the pH pretty constant, okay? So lots of times these buffers are what we call weak acids. And so I actually don't like the way this equation is written. When I write this equation, I write it like this. H2CO3 is how I write this. And then HCO3 negative plus H plus. Okay, this is more familiar to me. So, but can you see that I just rearranged stuff, guys? This is the exact same thing. So what is this? Well, this is called carbonic acid. And then this is called carbonate. And then of course, this is a hydrogen ion, which would be an acidic solution, right? So you have instead of an OH, we have a bicarbonate, bicarbonate. So instead of an OH, we have a bicarbonate ion, but it's functionally the same thing, isn't it? We have a negative ion and then a positive hydrogen ion. So if we had a lot of these, we'd have a base. And if we have a lot of these, we have an acid. So buffers are really important cellularly. And sometimes I like to throw in things that are actually examples from like medicine or biology, just so that you understand why we study some of these things. So if you are interested in nursing, you might be interested in things like ketoacidosis. You guys may have heard of that. You also might have heard of something called lactic acidosis, right? These are both forms of what we call metabolic acidosis. What does that mean? Well, these are processes in your body that lead to a lot of hydrogen ions circulating through your bloodstream. Ketoacidosis occurs when you don't eat any carbohydrate and you're getting all of your energy from fat. As you break apart those fats, you end up creating something called ketone bodies that are then metabolized into acids. Lactic acidosis is the result of, like, if you do a lot of exercise and you go into an anaerobic phase, we'll talk about that when we get to cellular respiration, you end up generating something called lactic acid. So in both these situations, you have acids circulating in your bloodstream. Well, let's look at this equation one more time. I'm going to rewrite this equation right here like this. Let's say you're in a situation where you have dumped some hydrogen ions into your bloodstream. Now your bloodstream has a little bit of this carbonic acid sitting around in it. All right, so we're going to have H plus plus HCO3 minus, and you've got your H2CO3. So I have reversed this equation, okay? That's all I've done is I've taken this equation and reversed it because I think it's going to be easier for you to see how buffers work if we do it this way. So in your bloodstream, you have some of this carbonic acid. And notice I've got these arrows going both directions. That means that this reaction is reversible and can move back and forth. Well, remember how I said buffers act as sponges? If you dump a bunch of extra hydrogen ions into this system, what's going to happen is it's going to drive the reaction in this direction. It's going to cause the hydrogen ions to combine with these bicarbonate ions, and you're going to end up with extra carbonic acid, okay? So having this around means this buffer can absorb some excess hydrogen ions that are generated during keto or lactic acidosis. And what's really cool is we could actually draw out this equation chemically a little bit farther. You can break, if you end up making a whole bunch of extra carbonic acid, because you have dumped a bunch of hydrogen ions in here. So these are combining and you end up with a high concentration of carbonic acid. This can be broken down into H2O plus CO2. What happens to CO2 in our bodies? We blow it out our lungs, don't we? So this is a really effective system for getting rid of excess acid in our bodies, okay? So when you think about a buffer, think about it as a sponge that's able to absorb extra acid, okay? And it can then convert it to CO2. What do you think might happen if you put in something like a strong base, which is an NaOH situation? Okay, that's going to generate a bunch of these hydroxyl ions. I've just told you what's going to happen if you added a bunch of positive hydrogen ions. What if we do the opposite and we add a bunch of negative hydroxide ions? Can you predict what might happen in this system? That would be a really cool way to check and see if you understand this. And again, it's a question that I'm very willing to answer for you on Friday. But I think the best analogy is to think of buffers as sponges that help to maintain pH, okay? All right, let's switch gears. We've talked plenty about water. Let's switch gears and talk for a minute about carbon. So that is this guy here, okay? We often say that life on Earth is carbon-based, and the study of the chemistry of carbon is called organic chemistry. And many of you will be taking that as part of your course of study. And you'll also hear horror stories about that class, but let me tell you something. I really enjoyed organic chemistry even better than general chemistry. All organic chemistry is that it's a discipline within chemistry that's devoted entirely to the study of carbon-containing molecules, okay? Now, the thing that I want you to know about carbon is that it is versatile. When you see carbon, the reason that it is a big, fat, hairy deal is that it is a versatile molecule. Why do I say that? Well, let's go back and draw out our little diagram. So we're going to have 6 protons, 1, 2, 3, 4, 5, 6. I'm going to put pluses here. That also means we have 6 neutrons because we're going to say that this is not an isotope. All right, so we got to have 6 electrons to balance out those 6 protons. So here's 1 electron, 2 electrons, and then moving back out. 3, 4, 5, 6. Okay, so you guys know that the second shell and then every shell thereafter wants to have 8 electrons. We only have 4. That means that we have 4 parking spaces available for bond building. And so what that means is there's lots of places that carbon can engage in bonds with other atoms, and therefore it makes an excellent molecular skeleton. So here we go. Carbon is an excellent molecular skeleton. And these are just some images of some different carbon compounds that I have for you here to give you some idea of the versatility of carbon. This is something called octane, okay? It is a hydrocarbon like fuel for your car. And you can see that what you have here is carbon. And then again, we've got 1, 2, 3, 4 bonds because we've got 1, 2, 3, 4 parking spots for carbon to make bonds. And this is a nice hydrocarbon chain, okay? Lots of energy stored in these bonds. Here we see that carbon has formed a ring. So this carbon molecule here has got a bond with oxygen. It's got a bond with another carbon group up here, bond with another carbon, and a bond with a hydrogen. And you can see that it's able to form this ring structure. Here's a glucose. So carbon is an excellent molecular skeleton. Think of it as a Lego system or a, I don't know if you guys know what connects are. That was a big toy when I was a little kid, but it was just this really cool construction toy where you could stick things in. So just think of this as a great Lego system, all right? And I have here for you these functional groups. I don't want you to learn these functional groups right now. We're going to talk a little bit about some of them when we talk about amino acids, but I just kind of wanted you to see some of the things that you can stick onto a carbon skeleton and that's going to give it different functions. So like here's a central carbon and we have what's called an amino group, this NHH group. It acts as a base. So if we stick an amino group onto a carbon, now we have created an amino acid and it acts as a base. It tends to attract a proton. Here's another one. Here's a carboxyl group. If we stick that onto a carbon skeleton right here, it's going to act as an acid, okay? And so I don't expect you to learn these. We're going to talk about a few of them when we get to our discussion of amino acids, but I just want you to recognize the versatility here. Like here's an OH group stuck onto a carbon. This forms an alcohol. Here's a phosphate group stuck onto a carbon, okay? When you have more than one phosphate linked together, it stores chemical energy. Think about ATP, adenosine triphosphate is 3 phosphate groups stuck onto a carbon, okay? So just know that carbon is really versatile. We can stick different things on there to change the functioning of molecules, and that's why life is carbon-based. One of the things that we are going to see in moving forward in our next few chapters, we're going to be studying carbon molecules as polymers, all right? So what is a polymer? All right, let's break this word down. A mer is the word root for unit. I can't remember if it's Latin or Greek, but it means unit, and poly means many. So a polymer is a molecule made up of many units, and it's a carbon-based molecule made up of many units. It's usually made up of individual units called monomers. Mono means one, okay? So in a polymer, you have a bunch of monomers that are linked together to one another, and each of these monomers is a carbon-based molecule. So I'm just going to write the word carbon. Okay. So you can see I've got a monomer, 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 and I've linked them together. Now, how do we link these monomers together to form polymers? Well, it turns out that these individual carbon-based monomers often have OHs on one end and H's on the other. And so you can link them together by taking away an H and an OH, which you guys know forms water. So if we take the H on one end and like got H on this end and an OH, let me make this clearer. Okay. So you see how you have an OH on this end and an H on that end? Let me draw that here. We have an H on one end and an OH on another. And if we want to link them together, we take those off. That gets lost as water and a bond is formed between these two, okay? So we actually say that polymers are formed through something called dehydration synthesis. And I'm gonna abbreviate a little bit. Y'all can, because I've got this stupid tablet, dehydration synthesis. Or another way of saying that, and your book uses this terminology, it is a condensation reaction. All right. Now, if we want to break these apart, let's say that we have our little polymer molecules that are, remember, each of these is carbon-based, and we want to break these apart, what we will do is engage in a chemical reaction that sticks these hydrogens and hydroxyls back on. Okay, we take a water molecule, break it apart, put a hydrogen here, put a hydroxyl group there. And so what we've done is we've used water to break it apart. So we've used hydrolysis, hydro meaning water, and lysis meaning to break. All right, so why am I bringing up all of this polymer nonsense? Well, because remember, we're talking about carbon, we're talking about carbon molecules, and we're talking about how versatile carbon molecules are. And our next unit of study is going to be all about looking at these polymers, these giant carbon-based molecules. Some very famous polymers are things like amino acids. I'm just going to write AA. Amino acids are the monomers that will link together to form polymers that you know as proteins. And again, these are carbon-based molecules, okay? We also have these right here. This is a carbon-based molecule. This monomer here happens to be a nucleotide base that's going to be part of a polymer called a nucleic acid. In carbohydrates, let's look at this. We have each of these, and when you see a drawing like this and there's nothing written there, then we understand that these corners represent carbon molecules. So that's a carbon molecule, carbon molecule, carbon molecule, yada, yada. We have a carbon molecule based monomer right here, and when we link them together, we get a polymer that's called a carbohydrate. And here we have a polymer of carbon molecules that are linked together. These monomers are linked together to form lipids, okay? So the reason I bring up polymers and carbon is because our next unit of study is going to be talking about these individual polymers that are so very important to our understanding of molecular and cellular biology, okay? And remember that polymers are formed through condensation reactions, and they are broken apart. If I wanted to break these apart into individual sugar molecules, I would use hydrolysis, or the power of water, to break them apart. All right, the very last thing that I want to mention to you today in our little discussion of chemistry has to do with how we got here with these large polymer molecules, okay? So you guys know that in your body's made of proteins, your body is made of nucleic acids, carbohydrates, lipids. All of these things go into the molecules that form biological organisms. So how did we get here? Because it turns out that we believe that in early Earth, the sort of chemistry of early Earth was some very simple molecules. And so one of the questions that scientists had for a long time is, how do these biological molecules form, these biological polymers form? Well, in 1924, notes down here, guys, 1924, there were two scientists that independently posed this theory called the Oparin-Haldane theory. And what they thought was, well, what we think happened in early Earth was we think that we had some very simple molecules. This is methane. CH4 is something called methane. So there's some carbon for you. This is ammonia. This is just hydrogen gas, and this is water. And these were very simple molecules, right, on early Earth. And what they suggested was that early Earth was a high energy environment. There was lots of like lightning and things like that. And they hypothesized that the conditions, the high heat of early Earth as it was cooling down, the lightning and the electric energy of early Earth might have created an environment whereby some of these things, there was enough energy to cause them to link up to form some polymers. And so that was their hypothesis, but it wasn't actually tested until 1953 by a guy named Stanley Miller in something called the Miller-Urey experiment. And let me show you how that worked. What these folks did was they had a flask with some water in it. So you've got heat, we've got boiling water. And what happens if you boil water over a long period of time? Well, it's going to form water vapor. And you can see what their experimental apparatus looked like. They heated up the water vapor went through some glass tubing, and then it passed through a chamber where they had some ammonia, some hydrogen gas, some methane, which is also a gas, and their water vapor. And they were able to pass electricity through there to simulate lightning, okay? So we have heat, which is a condition that we believe existed on early Earth. We have lightning, which we think existed on early Earth. And so then at this point, we're gonna let the water cool and condense. Water droplets are going to form, and then we're going to start this cycle all over again. And they let this go on for 1 week. And at the end of 1 week, they analyzed what was in the flask to see if any of this, like if the water vapor that went through this highly electrocuted chamber picked up any of these molecules, and did it form any polymers? And guess what? The answer is yes. So some of the things that they found were things like glycine, which is an amino acid, formaldehyde, hydrogen cyanide. And so the idea is that the conditions of early Earth, all that we needed was a high energy input and some very simple molecules, and we see the beginnings of some organic polymers beginning to be formed. So this is our theory of chemical evolution, and it's the beginning of how we think we got to where we are with cells and the cell theory. All right, so that brings us to the end of chapter 2. I'm going to go ahead and stop this video. Remember that chapter 3 will also be posted, and at the end of week 1, you need to make sure that you've watched 1, 2, and 3 chapters worth of video material. All right, talk to you on Friday. Bye-bye.